So start already. So so today what I'm gonna try to cover, so try to be broad, and then cover most of the approaches for like making transgenic animals. When we see it, the jargon words, the more transgenics, but then sometimes we also use very specific meanings of the transgenics. So you will see that I probably use those words interchangeably. But generally speaking, it's like certain genetic modification in the mouse genome. So maybe, thank you, Lydia. So where should I go to minimize that? Okay, and then that goes to very top here. And then going forward is here. All right. Um,
Might move the mouse off that. Get it to respond.
either that.
Oh,
or-
no. The laser is on, I think the arrow should work.
I think
Alright,
because you're
okay.
highlighting.
Because it was right.
It was
as
highlighting
in-
over top of it, yeah.
So I will divide it, so today's presentation to, generally speaking, four different parts of it. First talk about, so why we want to make the genetic or modified mice, or what we can achieve with this approach. And then, so in terms of the type of modifications, so that will also go, so like what kind of genetic modifications, so different categories. And then the approaches to generating those mice. And then finally, I would give two different examples. So actually, it will be the more complicated examples. So illustrate all these different approaches we talked about, and how in the end to achieve what we want to do every day. So what can we get with genetic modification? So we can so first of all, if you want to introduce a foreign gene to the mouse genome, right? You want to express a reporter. We'll come back in the next slide about what do we mean of a reporter. So, or you want to express a gene from a different species. For example, you want to make a human mouse, right? To directly use that to study human diseases. Or you want to express a gene that normally the mouse genome doesn't encode. So on the human genome, so you can put it there. So so that's not exactly the same as when we say humanized mouse, because that, in terms of the generating of the more the human gene forms of the coding sequence, everything. And then you can also express a toxin. For example, so people who, so for example, Ariel Lab before had used, and I think Lydia also used it before, right? So the DTA mouse, you express a tetrodotoxin. So subunit A, that's what DTA stands for. So you express a toxin there so that you can kill a cell. So that's cell ablation. So you can study not a gene function, but you study what the cells do during a process you study. And then more often that this is used is we want to use a to express pre-recombinants. And so the importance of pre-recombinants is to generating conditional knockout. So as mentioned here, and then that's one thing I would illustrate towards the end of the today's presentation. So that's introducing foreign genes that's normally not in the mouse genome. Another thing is you can also modify a gene in the mouse genome, an endogenous gene, but you can make it express in an ectopic place. For example, a gene normally expressed in the renin cells, now you want to express in podocytes, for example. So this gene normally you will never detect it there, but if you want to see whether it has effects there, or for example, there is human patients, then this gene is ectopically, so expressed in a different cell type, and we want to study what effect that expression has, then you can make that in the mouse. So using the mouse homolog of the gene, right? So that's what ectopic means. So it's not in where it's endogenous locus tissue expression. or you can express the gene, like you can, overexpress it, so you push the level by giving it a very strong promoter driving its expression, so not using this endogenous promoter. So other ways to do it is so this is also for your gene of interest. You can express it as a fusion protein. So now that you put a tag, you can put it as a GFP tag. So, or you put a tag like, so like, for example, the flag tag, so that people can generate a mouse, and later on they can use it to do immunoprecipitation for biochemistry. So, or you can modify that gene so that it will express a disease form of it, right? So, like so we have all these things about the human patients have the mutations. And then you can modify that to change it. And then so now you express a mouse in its, so either, so now it not express the wild type, but actually express that mutant allele, and you can examine how that affects its function. So these are more like so you see like all this above is more introducing expression. So it's like more like almost like a gain of function type of approach. But there's also we can create loss of function alleles with so gene modification, and that is gene knockout. So it can be a street knockout, so it can be a conditional knockout. Okay, so those are the things one can achieve with modifications. So one thing I want to illustrate, I just think this picture really explains one thing and shows, illustrates how one can achieve with this kind of genetic modification. So, and then this also explains what is a reporter. So this is, so currently probably one of the most sophisticated reporter called confetti. And then so this is actually a human brain. So reporter is proteins that we use to actually tell us a gene function or label a particular cell type. And for that, in the past, we use legacy. So people have so like initially the field is using legacy, so you can do beta gal staining and then you see those blue ones. So if you read papers, before 1990s, you'll see a lot of this legacy reporters. But more and more people use the fluorescent proteins, GFP, YFP, CFP, RFP. Tomato is so similar to that. So so this is one that people use, that this construct, which actually has all 4 different reporters. And then by Cree-mediated recombination of all this, is all random recombination, you can generate cells that are labeled with different fluorescence. And then so not only, right, we can, while looking at this picture and
That's vegetable, and this is mushroom. Something.
Thank you.
Oh, sure. Thank you. So while we digest food, we'll also digest the picture. Okay, so what is good about this? So there are so like a reporter, especially for these brain people, because you have all exons, all tangled, not tangled, but this very dense area, like in this in the cortex region, cortical region, right? But with this, they actually can trace like which cell, which exon, comes from which the neural cells. Okay, so actually, I think at one time I saw the person who was doing it, he was having this images, and on the computer, like manually, that was tracing vertical sections, the sections and go like that. But at least it allows you to do that. So for us, for example, so almost lab people also use this competing mice, right? And then you can see like the recruitments and then see all those different along the arterioles of the smooth muscle cells, recruited really expressing cells, and you can see them in bands. So and then so when you have a reporter, so this really labels the cells. Another thing it tells you is when you want to so for example, you knock out a gene. So if you have this reporter there, it also labels which ones are your knockout cells. So you really can look at, and then for example, this days when we're doing single cells, then you can sort those cells out. Those cells will be, you sort those cells that are only the knockout cells. So reporters can really help us to do multiple things. In addition to being so visually. So already, so that is already covered. So what I want to do next is to say, so okay, we can do all these different things, modifications, but when you get down to technically, so how the mouse genome is modified, there really, so in nature is this three different types. So random integration, knock-in, knockout, knock-in, knockout sometimes happens simultaneously. So what do we mean of random integration? And so this random, it really means is this piece of DNA fragment you're introducing into the mouse genome is inserted into the genome in a random place. So we don't have control where it lands. And so because of that, so this you can use it to do reporter gene expression. So all this you introduce a foreign gene, you can ectopic expressive gene. So all this, if you want to achieve this type of goals, you can use random integration. So and then how do we do it? So technically we do it by pronuclear injection. And so that is the slide I'll go just walk you through this one. So you start with one mouse. That's so you get it super ovulated just to give you a lot of the one cell stage, the fertilized egg. So at one cell stage, before the male and the female pronucleus fuse to become one common nucleus. So at this stage, then you just inject your foreign DNA into the pronucleus. So like our facility, so UV transgenic facility can offer this approach. So after injection, then all these injected fertilized eggs then gets transferred to a surrogate mother. So and then from there, so not all the eggs when you inject will have the DNA insertion into the genome. So after that, so the pups born from this surrogate mother, some of those come from this one cell infect. So this one cell becomes the embryo grows in the mom, right? And then that becomes the offspring. So this offspring, some of those are eggs, even though injected, but the DNA didn't incorporate into the genome. But some will have that incorporation. And so now you have your founders, and then you screen your genotype for your founders. And once you get a founder, you need to cross another time, breed one next time to see whether it goes germline so that you can establish it. But there are times we just do G0 analysis. We just want to see whether this gene has a function. It's a very quick assay. Then people can just, you don't do even this step. Then you just analyze this once. You can do that. But if you want to generate a line so that the lab can continue to study that, then you want to cross this to at least get, so not even every one of this will give you germline transmission. That's what we look for, so that you can establish the colony. And for this kind of approaches, so the nature of this type of injection. So first of all, the location can be all random, and then you can have even multiple insertion sites, so that one founder mouse, when you cross, it may segregate, chromosome 1 may have one, chromosome 20 may have one, and then when you cross, then you separate those ones. And then also, even in the same insertion site, you could have competitors. We do not even know how many copy numbers it has. You can have a single copy, you can have multiple copies, all like the intended matrix. So the good thing of this approach is it's very simple and fast. And then you basically, so a lot of times when we make this, before we do it, our construct will test it in a culture, in a cell culture, right, to show that it really works. And that construct, basically, you don't need to do any modifications. You can just go directly to this. So it's very easy to adapt. But the problem, on the other hand, is there's this copy number variation. There's also position effect. So in our genome, we have hot spot and cold spots, right? And then not only that, so your insertion site can be close to heterochromatin. And then your expression level, so position effect variation, that is what we talk about. And then so even if the same copy number, depending on where that insertion happens in your genome, the expression level can vary. So together with copy number and then insertion sites, you have the same injection of all these independent lines, their expression level can all vary. Okay, and then you could also insert into a place that disrupt endogenous gene expression. So when you see it, you get an effect. It does not necessarily mean it comes from your gene, your genetic modification. So because of that, you have to screen, you at least have to have 2 mutants, most likely better to have 3, if they all give you the consistent phenotype. That's the phenotype that's more likely to come from your introduction of this foreign gene. So this is particularly important. So like if it's a reporter, like all you care is whether whether it's a very sensitive reporter, it's a cleave with whether it functions, immediate pre-mediated recombination very efficiently. But if you're doing something of expressing your gene of interest, so then the level, you may get to different levels that doesn't even tell you what you find analyzing those animals. If you analyze just random, it may not even faithfully reflect what's the function of the gene or the mutant form you're studying, right? So, and not to say the contribution, if you happen to knock out gene X in the genome, what you observe is a combination of your expression of the gene you introduced and then the knockouts of or knockdown of that gene. So that is the pronuclear injection. It's so pros and cons. So because of that, then people developed another method is to do knock-in. And then, so in addition to that, knock-in also have its own advantage. So what knock-in means is you actually introduce the DNA fragment in a defined locus. And then in this, really, it's two categories. One is what's commonly used, ROSA 26 locus. And then the other one, of course, is your gene of interest. If you want to modify your own gene, then of course you have to target, if it's renin, we target renin, if it's APR, we target the APR 1B7, CREAT2, APR 7, right? 1B7. But so ROSA 26 locus, this is our most famous locus in mice and also in rats. This is the locus. A lot of time when people say knock-in, so that is the one. And then when you see a mice, then they will really see ROSA 26. So for example, MTMG is a reporter mouse line that we use a lot. So it's reporter MT, so membrane tomato and a membrane GFP, right? And then that is a mouse, that's the reporter construct is actually knocked into the ROSA 26 locus. So what's so good about the ROSA 26 locus? This is a locus that is not essential for mouse development or life. So you can feel free to insert anything that's not going to generate the effect we talked about just a few minutes ago, right? And then another thing is this is a relatively it's an open locus, so you don't have to worry about heterochromatin effect on gene expression. And also that's a locus that's pretty much open in all cell types. So there are only a few cell types that's not perfect, but most of the approaches have been used. And then so one can either use the ROSA 26 itself as a promoter. So that is an intermediate level of promoter. But one can also just clone in your own promoter of interest. So for this, you can achieve similar things to what we just said, use random integration method. You can still use it to express a reporter as MTMG, we can clone it into the ROSA 26 locus. You can express genes from different species. So there is also, for example, DTA mice. There is a transgenic DTA mice. There is also ROSA 26 mice. So that people who target the ETA coding sequence into the ROSA 26 locus. So you can guarantee a more stable expression. And then so you can also make a CRE there. So the thing is then you have all these ones, you can also ectopically express a gene by putting the entire coding sequence together with your promoter of interest into the ROSA 26 locus. So all this you can do it. So let me go back. So in addition to going to, so that's talking about into the ROSA 26 locus, but you can also target your own gene functions, right? So in that way, you can drive the gene expression with the endogenous promoter. So you maintain the expression level and also the cell type that express it. With this, especially if you want to express a disease variant of your gene interest. So if you target into your endogenous locus, that's called knock-in, then that disease allele is driven by the same promoter as the wild. So that will be really mimicking what's happening in the patients. And then so for this approach, actually CRE is something that we use a lot. So there are plenty of CRE. So these days, when you get a CRE from people, you can ask that question: is it transgenic or is it not? So that's what I'm saying here. Transgenic, we use it in two different terms. We use transgenic broadly is equal to saying genome modification of the mouse, but we also very narrowly when we talk about it is actually more that random integration type. So that's a transgenic, supposed transgenic. So versus a not-in, so you want to express a creed, okay? So do you want to use, for example, nephrin or rin-1D creed? You actually can knock the creed coding sequence into the rin-1D locus, for example, then it will be driven by the endogenous rin-1D promoter. So the expression, so you don't have to worry. It will faithfully recapitulate where renin is expressed. So you will guarantee use that when you knock out your gene, it will be knocked out in the renin lineage. So that would be it. So that's one example to see here. So even when we are making a cree line with knock-in, there are also two different ways of doing it. One is you knock in at the C-terminus bit. So with this, you do not even disrupt your gene of interest expression. So in the end, so then you here you put the RAS, or this case people do T2A, right? So and then you do it so you can have your protein still being expressed, and then it will come with another translation to make a cree protein. It's not fusion protein. These will be two different peptides. So this way you won't create a knockout of your gene. So I think this, yeah, so that gets, let me see, I missed one. Sorry, let me see, there should be another page. Oh, okay, so then I probably use that to talk about already. So then the knockout approach, so that is also, so knock-in in a sense, it's not how to knock-in in terms of genetic approach to create that allele is similar in that you actually, instead of insert something into your locus of interest, you actually now delete something of of your at your gene interest. So for example, here you have a gene that of multiple exons, which is more common in the mammalian genome. So then you delete one exon or a couple of exons, those are critical exons, then you generate a knockout because the protein is either not a peptide that's very truncated to the point that there's no function. So, or you can create a knock-in, knockout. So that's what I'm saying, knock-in, knockout can go hand in hand. So the example here is actually, if you introduce cree, you knock cree into your gene of interest. So again, I'm just saying if we use rin-1D as an example, right? So that's simply knock into the rin-1D locus. But when we do this, we simultaneously, if we do it in this scheme, we simultaneously also knock out renin, because you have the polyadenylation tail here. So this locus will only translate. So even though if you look at mRNA, you have to see cree and renin mRNA. But if you look at protein, then there won't be renin protein because translation stops here. So that's what, so one can create knock-in, knockout. You can use this, so just by where you insert it, where you knock in, you can have just purely knock-in or knock-in. So the one last thing of when we use this type of the like knock-in, knockout, so it's creating conditional knockouts. So do everybody know what conditional knockout means when with this term? So conditional knockouts versus straight knockout. Straight knockout is from the one cell stage, you lose that gene. The gene is knocked out. So all your body, every cell, from the day you were, like the mice is conceived, that's it. So the gene is completely gone. Okay, if the knock, so by design. Conditional knockouts is in a way that the gene will be deleted only where you want it to be deleted, and then when you want it to be deleted. So that's why it's conditional. All right. So again, so that I will illustrate later on. So, but even for this, to make a conditional allele, we have to knock into the gene, the locus, at least minimum 2 logs P sites, so that later on pre-recombinase can use it to mediate gene deletion. Okay, so how that's achieved? So traditionally, so people use ESL targeting for knock-in and knockout, conditional knockout. All this is the same technique. So these days, and then today I won't cover that because Sylvia will talk about, right? So CRISPR technology. So with CRISPR, you can do all these things, the random integration, the knock-in, knockout, is all it's all feasible with that technique. So this will be talking about what it has been done, and then they are still being used, especially for the pronuclear injection. And I have to say, even for CRISPR, you still, even for introducing CRISPR, you have to do pronuclear injection. So traditionally, when you do ESL targeting, so you first have to have a targeting construct. And that construct you introduce into a ESL culture. And then so the foreign DNA gets introduced into the ESLs, and then you have to select for the positive ones that incorporates into the genome. And that actually goes with the homologous recombination. So it will only get your DNA sequence will only go into your location, whether that's a ROSA 26 or that's where the gene you want to modify. So after that, once select, and I can tell you, it is like this bed of internal the efficiency. So you have to, you grow 10 to the 7 cells, and then in the end you screen for the single cell colony kind of screens, and you get a couple of lines. So then in the end you grow up the ES cells that is targeted, and those cells then gets injected into the blastocyst. So this will be later stage than what we talked about in the random integration. That's just one cell stage. That's the beginning of life. But this is already so 4 days, 3-4 days after that. And then so they have this, so the embryos grow to the blastocyst stage. And the cells is injected into this empty cavity, and then gets incorporated into this mass of cells. And then this, so again, we have to inject a lot of this blastocyst. And then again, this will be implanted into the surrogate mothers. The pups born from the surrogate mothers are derived from this blastocyst, but they will be mosaic because this not only contain, these embryos not only contain the ES cells you inject, they also have their own ES cells. So that's why it's drawing all like this. It has mosaic. So and then so you look for each of those mosaic, you look for ones that has more of the coat color representing more contribution from your injected ES cells. And then those ones, then you do another cross and then look for ones that carries more your coat color, the coat color of your ES cells. And those ones are most likely the ones that completely come from your injected ES cells. And then, so from there, you also need to do then the go to germline transmission. So it will be another round of cross to make sure that you all go germline. So those are the things. So founders are mosaic, and you have to screen for germline transmission. So now those are really all the general things about, like what are we talking about when we are saying transgenic animals? How do we get it? So so I won't touch on all the details, like what we do it. What I'm going to do now is to use two most complicated kind of schemes to illustrate what all these different techniques when it comes. So first of all, so it's a conditional knockouts. Okay, so and then so when you have a conditional allele, so as we say then we knock in the two LOXP sites to flank one or more essential exons. So that's why a lot of times when you see the lab writing it, they write F the flux. So it's a flanked, so by LOXP, so this word that is coined flux. So once you have this, you can create all kinds of gene knockouts. You can cross with a Cree that's expressed from day one of animal life in all the cells. For example, SOX2 Cree that has been used, so people use in the lab, right? So that way you generate a straight knockout. Okay, so the gene, so it's equivalent to when we do say knockouts that completely remove the essential exons. Or you can cross with a Cree that's driven by your promoter of interest. For example, if you want to just remove it for the renin lineage, then you cross with the RIN1D Cree, so that this gene will only be removed in the renin lineage, and everyone else are all normal, no type cells. But for this type of approach, from the moment renin is expressed, so embryonically, then this gene will be knocked out. Okay, and then so that sometimes it's good for people who studies development. But if you want to look at postnatal adult life, so like handling disease, drugs, all these type of things, then that potentially can be complicated by the early developmental defects. And then one can cross with a tissue specific inducible. So what do we mean by that is that the Cree normally, even though it's in the genome, it's even in the right cell type, it's expressed in renin cells, for example, but it's not functioning only when you put in the inducer. So what do we mean of that? Is that, so one common approach we use is tamoxifen inducible. So in this case, you have a Cree, so your renin, for example, I'm just using renin because most people are not, so we all know renin, what it is, right? So and we are using that. So you renin promoter, for example, drive this gene, this protein called Cree-ERT2. So this is a Cree recombinase fused with a estrogen receptor. So normally, without tamoxifen or estrogen, then the Cree-ERT2 actually is expressed, but it's localized in the cytoplasm. But we all know what we want the Cree to do is to recombine the genome, the DNAs, right, to recognize the two LOXPs to induce recombination. So in the cytoplasm, it's not going to do anything. So it doesn't really matter that it's expressed in renin cells, but it's not functioning until you decide, you determine this is the time I want it to work. So then you inject tamoxifen or you provide tamoxifen. So you give tamoxifen to the animals. And then when the Cree-ERT2 protein binds to tamoxifen, it translocates into the nucleus, and where it will see the LOXP in the genome and then induce recombination and therefore knockout. So this is why you can control both spatially by using which promoter driving the Cree, but you can also control temporally by when you give tamoxifen. But this is an irreversible change. Once you give enough tamoxifen to induce this recombination, the genome is modified, permanently modified. So the genes deleted in all the cells and their progeny that express the Cree. Okay, so so another way to do this, so you can also have spatial and temporal control, is the TET on, TET off system. So for this, a lot of times we more use this as a gain of function approach. So the one we talked about just now, tamoxifen inducible Cree, is most of the time we use it as a loss of function. So it's to knock out the gene. But use this, you can drive gene expression. So you can ectopically express gene. You can express all this, like modified form, but you can also use it to do gene knockout. And I will show you the strategy for doing that. So what's most different between the tamoxifen inducible CRE versus the TET system is that the TET system is a reversible system, and I will show you in the next slide. And so the elements we're going to use, we need tissue specific TTA and RTTA lines. So we need, so in some schemes, we use flux RTTA and tissue specific CRE lines. So that would be just like we talked about the conditional knockouts. And then we can also have TRE CRE and conditional knockout. So let's see all those kind of scenarios. So what is TET off? So TET off is, again, you will have two components. One, you have the gene you want to express is cloned after the TRE, so the tetracycline responsive elements. So normally, and then you have another mouse, so that actually express TTA protein, and driven by the promoter of your interest. So I'll just make this up, say that we have a 1D promoter driving TTA protein expression. Okay, so when you cross these two animals together to drive to get this compound lines, so from the beginning in the cells that express really, the TTA will be expressed and then binds to the TRE, and that will turn on the gene expression until you add. So here it shows tetracycline, but we more use doxycycline, which is a tetracycline derivative that's better than tetracycline. So when you give tetracycline, then tetracycline binds to TTA, and that competes with the binding with TRE so that it gets off the promoter region. So now this protein stops from being expressed. So that's why TET off. When you add TET, gene expression is turned off. And then alternatively, later on, people also developed TET on system. So where you actually, instead of say like, again, I'm making this up. So you have a Ring1D promoter driving RTTA expression. And then you have your gene interest. So that's the same construct as we talked about before. And then so normally, this RTTA by itself cannot bind to the TRE. And therefore the gene expression is turned off. So it's constantly off until you gave the animal, you feed the animal with doxycycline. And when that happens, the doxycycline binds to RTTA, and then this compound then binds to TRE, and that turns on gene expression. So that's why it's called TET on. Only when you give TET or the doxycycline, then you turn on gene expression. So both of this has good and bad. It really depends on what you really want to do and what's more convenient for you, right? So if you want to study the animal like 1 year, 8 months old, you want it to be on, you want it to turn on at that time, right? So if you do TET off, you're gonna have to feed this animal with doxycycline all 8 months. That may not be something you would love to do, right? But if a TET on system will be great because you don't need to do anything, they just learn. And then at the time, then you give doxycycline, so you turn it on to express it. And then there's also practical considerations about which system is more key kind of things. But what I want to really emphasize is this both TET on and TET off system is actually a reversible system. If you look at it, it didn't change. So the tetracycline, when you give it, it didn't change the genome. So you can even, for example, you give TET doxycycline for like 20 days, and then you remove it. So you first look at, like when you knock out this gene, or when you ectopically express this gene, what effect you get it. And then when you remove it, whether like the phenotype will be reversed, right? So one has that flexibility that's not seen in most of the pre-LOX-P system, because pre-LOX-P, once you did it, you modify the genome. So even after that, you stop injecting tamoxifen, which that's what the standard approach is anyway. But it doesn't, it's not gonna make any difference. What's deleted is deleted, it's not gonna come back. So that is the difference between these two systems. So one can also use the TED-on system to achieve what we do with the inducible Cree. So for this, you need 3 elements. So we need a fluxed RTTA allele. We need a tissue-specific Cree. That's just like, that's the most widely available tools we use. You know, we have Ring1D Cree, you know, so if you can have a smooth muscle Cree, you can have a nephrin Cree to label the podocytes, you can have a Cree that labels the endothelial cells. So people have generated a large collections of tissue-specific Crees. So that is an easy one. And then you use the TET-O, that's another way to call TRE. So your gene of interest. And then when you have all this together, so your Cree, once they see this ROSA26 flux RTTA, it will immediately generate this. So the RTTA now is constant in the tissue and the cell types you want it to be. But that RTTA is not going to do anything until you give it doxycycline. And then you will turn on the gene. So this is a 3 component approach. And you will ask, why? Why bother? Isn't that the same as you just mentioned about the RTT, the TED-on system? It's all about availability. What the field already have, because you may not want to spend a year to make your own tools and characterize it and prove to the rest of the world this tool is behaving as you designed it to. So if there is already established allele, then that's great, right? So chances are, so this is why this approach, even though it's more complicated because you have to cross 3 different mice together, but in a sense, this is actually the more easier ones to get, because the ROSA26 RTTA, flux RTTA mice is a common mouse stock, it's freely distributable. And chances are you start, as long as you are not the first person studying in a very narrow field, chances are you find you historically in your own lab or your colleagues in the field already made a tissue specific. So the only thing is really you only are your gene of interest you're studying, you may make it. And chances are somebody else studying, for example, brain for kidney people, we always can look at the brain people, you know, they probably already made a line for us, right? So you probably already, you go around, you find all those components are already there, you know, you just need to get from people or from those common consortium. But the problem with the TED-on system is not only you have to have your own, you know, like the TED-O gene, you also have to have a tissue-specific RTTA. Obviously, that's a lot more rare than a tissue-specific Cree. So people usually do not make one like that. So that's why, even though this looks more like kind of clumsy kind of approach, but you probably even have a better bet in getting everything without yourself making a mouse. Okay. So the last thing, the last thing is that we talk about, so gene knockout, you can still use a inducible using the TED-on system to do the inducible Cree equivalent of things. So basically here, you have your RTTA driven by the promoter, right? And then you actually have a Cree that is a TRE Cree or TED-O Cree, so as people say it. So, and then you have your flux allele. So again, it's a 3 components once. So you determine when you give doxycycline, and that's when the Cree will turn on, and therefore it will go to the nucleus and then do the remediated recombination. And then the temporal control, of course, is when you give doxycycline. The spatial control is the tissue-specific promoter driving the RTTA expression. again, as we talked about, this is actually a much harder tools available. But there are times, so the field people has already made it. And then if nobody has made a CREAT2, and yet somebody in the field already made a RTTA, with the cell type you look for, then that's also useful to have. Because the others, your GM interest, hopefully somebody already made a conditional knockout, and then this will be a common cell mouse. So all these things. So a lot of this is really, which one we go, is really about available tools. You look around what's published and what's freely available, which one will allow you to achieve your goals faster and easier. But another thing, if you have the luxury of both of them, or sometimes you finally got all the three components or two components that you can do the study, you also have to consider whether the approach you can do, whether you can use it. For example, tamoxifen injection. But tamoxifen is estrogen, so it's a ligand for estrogen receptor. So male, females, or versus some other organs in our bodies are affected by estrogen. So that's something one has to consider. And doxycycline is an antibiotics. So if you want to study gut microbiome, you may not want to do that, right? Or so doxycycline could even affect mitochondrial function. So if that's your major things you're looking, you may actually, and then you say, oh, I have controls, but this is you not only doxycycline, but also your gene knockout. You may be looking at something of a synergy versus the control wouldn't even, allow you to control for that kind of defects. So one have to consider the availability and also your specific system. So your specific scientific question, whether it will be influenced by the drug, the inducers. Alrighty, so questions? Okay, so that we'll just, next time we will, so in a couple weeks, we'll hear the CRISPR?
It's next week.
Next week, okay, awesome, yeah, that'll be good, and hear all this, the new events of this. Yeah, alrighty, good, thank you. Yeah, yes.